Now that we have covered the introduction part, so let's proceed with the practical section of TR Rosetta. So to begin, please go to UniProt now, then select a protein of your interest. Here I select hemoglobin with this entry name P68871. Okay, then go to its sequence section. Um, then just copy sequence from here and return to our workspace and paste the sequence into this text box. Okay, so also select this input type. Here I use only this sequence, so I select query sequence as the input type. So we can give our email address so that results will be sent to this email address, and we can also mention the target name. Here I mentioned that as human HB. Okay, then click on the submit option. So our job has been submitted successfully with job ID TR161874, and we can track the job status here at this link. So the protein with the name human HB has been successfully submitted, and we can download the submitted file here if needed. Okay, and the process is estimated to take around this much time, and the current step is shown in this red color, and the other steps are it is now in queue in cluster system. And in step 1 it generate multiple sequence alignments. Okay, then step 2 is detect homologous templates using threading. So the tool checks if your protein sequence resembles the structure of any previously solved proteins, and this is called threading. The idea is to align your sequence onto these known structures or templates to find the best fit. It's like checking if there is already a similar building blueprint before designing a new one. If templates are found, they serve as the starting point for modeling. Otherwise, the tool proceeds without them. And step 3, it will predict distance and orientation distribution. This is the core of TR Rosetta. Using deep learning models, it predict the distances and relative orientations between pairs of amino acids in your protein. And these predictions are based on statistical patterns learned from thousands of non-protein structures. And this step essentially lays the groundwork for assembling our 3D model. And step 4 is coarse grain structure modeling. So once the distance and orientation predictions are ready, the tool uses them to build a rough 3D model of the protein. And this is called the coarse grain modeling, where the protein is represented in a simplified way. At this stage, the focus is on getting the overall shape and fold of the protein right. And the tool also ensures that the structure is physically stable by minimizing its energy. This means adjusting the structure to avoid unrealistic overlaps or strains. And in step 5, after creating the rough model, TR Rosetta refines it into a detailed all-atom model. And this step is called fast relax. It optimizes the positions of every individual atom in the protein. And it uses physics-based calculations to ensure that the structure is as realistic and accurate as possible. And this process fine-tunes bond angles, side chain orientations, and other atomic details. Okay, and step 6, it generates results page. With the refined structure ready, TR Rosetta generates a results page. This includes the predicted 3D model of the protein, downloadable data files, and additional information about the prediction. And researchers can use these results for further analysis like studying protein interactions or designing drugs. Okay, and finally, when the job is done, the system sends a notification email to the user if you give that email option there, and it ensures you don't have to keep checking manually. The email includes a link to access your results. Okay, So please note that this process may take some time to complete. So here it shows the estimated time required to finish this entire process is approximately 2 hours. Okay, so I suggest you to provide a valid email address, and this will enable you to access your result via a link that will be sent to your registered email address. Okay, so that's all about this TRO setup. Then we can wait for our results.